The first one is going to be Alana Tress, Associate Director of Patient Advocacy at PepGen. Then we're going to have Trisha Harden, the Director of Clinical Operations at Avidity Biosciences. Then we'll have Naz Dastier, the Clinical Development Lead of the Duchenne Program at Regenxbio. And lastly, we'll have Catherine Krieger, the Director of Patient Advocacy at Edgewise Therapeutics. And I believe Catherine is here, so perfect. So Catherine and I will be up on the stage and the other 3 talks will be virtual.
Thanks, everyone. My name is Alana Tress, and I'm here presenting on behalf of PepGen. I was really looking forward to being with you all in person today, but unfortunately, COVID had different plans for me, so I'm sparing you all of that, but it's been really a pleasure to be watching online. So I'll be going through mainly today our Duchenne muscular dystrophy program, otherwise known as PGN EDO51. I'll be going through some of the preclinical studies and data we've collected so far and let you know from all of that how we have ended up currently in the clinic. Here is our disclaimer slide. As Erin said, if you're looking to have any help with sleeping, I'm happy to send it to you. I think the biggest takeaway from this slide and the remainder of my presentation is that everything I'm speaking about today is investigational. Nothing has been approved by any regulatory authority just yet. So who is PepGen? We are headquartered in Boston, Massachusetts in the United States. We are small but mighty. We are about 60 employees and growing. And I'm proud to say now that we are a clinical stage company that has clinical programs both in Duchenne muscular dystrophy, which I'll talk about today, and also more recently, myotonic dystrophy type 1, otherwise known as DM1. And for both of these neuromuscular disorders, we utilize a platform that we'll call enhanced delivery oligonucleotides, or EDOs. You'll hear me refer to these throughout the presentation as EDOs, and I'll go through a little bit more about them in detail. So what are EDOs and why are we using them? Typically in the past, there's been something called oligonucleotides that have been used in exon skipping that you see on the left-hand side of the screen here, that gray nucleic acid chain that you see. And if it's not conjugated to something or doesn't have something to help it get into the cells, it typically doesn't. And if you look on the right-hand side of the slide here, what we've done is we've developed a peptide to connect to this chain. It looks like sort of that navy and light turquoise seahorse you see connected to it. And once that's connected to the chain, we've seen up to 25-fold higher levels of nuclear delivery. So getting into the cells and also getting into the nuclei, which we've learned a lot about this weekend, how important that is. So what are we doing with these EDOs? Like I said earlier, we're applying them to numerous neuromuscular disorders. So our first and most lead program is in Duchenne muscular dystrophy for people amenable to exon 51 skipping. That just entered phase 2 about a week or 2 ago, which we're so excited about. And then not too far behind that, we also have a clinical program for myotonic dystrophy type 1. And then additionally, preclinically, we're still studying other Duchenne muscular dystrophy programs. So and the exons for that are exon 53, exon 45, and exons 44. And again, this whole platform does utilize our EDO technology. So I really like this morning John's presentation talking about, if we can all assume we're 11th grade science learners. I really like that analogy a lot. And we created this slide to really set you up, hopefully a little bit better to follow the rest of our presentation. I'll be going through some of the animal data that we've collected so far about PGN EDO51, and with that data, how it sort of supported our thought process to then test it in humans. Humans are animals too, whether we like to agree to that or not, and eventually in the Duchenne population. So you'll see at the underneath each of these animals, so the mouse, then the monkey, or otherwise known as the non-human primate, and then the human, what have we learned so far about this candidate and how are we taking it into the future? So starting out with the mouse, we saw that for the mouse analog, which is PGN-EDO23, that we saw more exon skipping than earlier generation exon skippers. So you see here on the slide, the far left-hand side is that naked oligo. So that first oligo we saw with nothing attached to it, it's not going anywhere. There's a second out there is a second generation of oligonucleotides. Actually, some of these have been approved in the US. They do get some exon skipping in the mouse, but then we were really excited to see with our peptide and oligo that we saw higher levels of that. So we were really encouraged by that to say, hey, let's do some more studies, which the next study that we did in mice was doing repeat dosing. So what's happening to exon skipping? What's happening to dystrophin production if we did multiple dosing? And you could see here that with each dose, the number of exon skipping increased in the biceps. It also increased similar in the biceps for dystrophin production. So again, this has all been really encouraging for us to say, hey, let's take these findings and this investigative therapy into monkeys and see what we find there, while finding more out about what dosing could look like. And so we did a monkey study looking at exon skipping in multiple tissues, including the biceps, the quadriceps, the diaphragm, and also the cardiac left ventricle. And we saw levels of exon skipping, which were really encouraging, which is always promising in the drug development process as you're moving from the preclinical to the clinical stage. And so what we actually completed last year was a study of PGN-EDO51 in people living without Duchenne, otherwise known, you might hear elsewhere, called a healthy volunteer study. And so in this study, we gave them a single dose of PGN-EDO51. You'll see at the bottom of the screen here, that was in 5 milligrams per kg, 10 milligrams per kg, and up to 10 milligrams per kg. And then the end of this at day 28, so 28 days later from the dose, we did a biceps biopsy. And we were really encouraged here to see some of the data and action that we saw carry through from the mouse and the monkey into humans with exon skipping in the biceps. So now what do we do with all of that? We recently moved into the clinic, which we're really excited about. So earlier this year, we got a no objection letter from Canada to begin our Connect 1 trial in Canada. The full name of that trial is Connect 1-EDO51, or you'll often hear us call it just Connect 1. And then we've also been working with other regulatory agencies to do our Connect 2 study, focusing on the Connect 1 study that is now open in Canada. It is a multiple ascending dose study, meaning multiple doses will be tested in this study. It's open label, so everyone that enrolls in this study will get the active drug. There's no placebo arm. And we're looking to get data for this study sometime next year, looking at dystrophin data, exon skipping data, and safety data. And it will be dosed once every 4 weeks based on, some of the preclinical data we've seen. We do have a site open in Canada, and we have a couple other sites that are soon to be open in Canada in the next coming weeks and months. Right around the corner from that, we also, like I mentioned, have that Connect 2 study that is going to be open in various European countries and the US. And that is a phase 2 randomized double-blind placebo-controlled study. So that means people, when they enroll, they will be randomized between whether they get Connect 2 or whether they'll get PGN-EDO51 or a placebo, and that's also a multiple ascending dose study. And we're really looking to find out from these two studies. We're excited that these will be looking at people, again, amenable to exon 51 skipping in the Duchenne population. And again, I just want to thank all of you today for having me, even if virtually. I know I've met with some of you in the past as part of our ad boards, and that's been so helpful for us. This is a picture of our summer event from this past summer. We had a little golf outing. If you have any questions for me in general, you can reach out to myself and my team at community@pepgen.com. If you are interested in learning about our clinical study and the details on it, we welcome you to email clinicaltrials@pepgen.com, and we'll make sure we get you in touch with the correct information. I'm also, I'll be on the Whova app for the rest of this conference as well, and you're welcome to reach out to me there. So thanks, everyone.
So thank you so much, Alana. That was very informative information. We're going to be moving on to Trista Harden. I know her slides are up, and I see her online.
Thank you so much. Can you hear me and see the slides okay? Wanted to check. Okay. Well, like Elena, I wish I was there with you in person as well, but really excited to meet with you virtually today. And thank you to Defeat Duchenne Canada for inviting Avidity to present. So yeah, my name is Trista Harden. I am a Director of Clinical Operations at Avidity Biosciences, and I'm the ClinOps lead for our Duchenne program. And I'm excited to talk to you today about our approach to exon skipping. So these are our forward-looking statements. Like Elena said, if you want to read them to fall asleep, that makes sense. So Avidity is a smaller biotech company, and we are located in San Diego, California, in the United States. We are focused on treating rare neuromuscular indications, and we are also a platform company. So we have what's called an antibody oligonucleotide conjugate, or an AOC technology. That we're using in all of our neuromuscular indications now. And so we have a cartoon on the screen here that shows an AOC. So the big part on the left is called a monoclonal antibody. And all of our AOCs across all of our platform drugs includes this monoclonal antibody. And the purpose of that is to target the transferrin receptor on muscle cells. And so the purpose of that is to bring the entire AOC into the cell. And then the red part that's on the right-hand side is what's really the business end of the AOC. And that is the part that's really designed to treat the genetic causes of muscle diseases. And in this case, for the AOC that we're talking about today, specifically for Duchenne. So I'm very excited to talk about our Explore 44 study, which is a phase 1/2 study of our AOC 1044 technology. And so we designed the Explore 44 study in collaboration with many folks. So we have spoken with patients and caregivers, advocates in the community, and also expert physicians and KOLs that work in Duchenne. And our primary goal for this study is to look at safety and tolerability of AOC 1044. This study is a placebo-controlled study, so it's randomized, double-blind. We do have 1 out of every 4 patients that are receiving AOC 1044 will receive placebo. And then the 1044 is given as an intravenous or IV infusion. So we have 2 parts for the Explore 44 study. Part A is being conducted in healthy male volunteers, and that is being conducted at a phase 1 unit in the United States. And then part B is specifically focused on Duchenne patients that have mutations amenable to exon 44 skipping. And we're looking to enroll about 24 patients in part B. And that part B is being conducted in the United States and Canada. So our current status of the Explore 44 study is we have cleared the healthy volunteer cohort, which allowed us to start enrolling in part B in our DMD 44 patients this summer. And so we have started enrolling in part B. We currently have 2 clinical sites in the United States that have been activated, but we're working really closely with additional sites in the US and in Canada to get open and activated for enrollment so they can start enrolling. Right now, we're projected to have our Canadian sites up and running the first half of next year. And we do have our study listed on clinicaltrials.gov. And then we also have a study-specific website that is here on the screen called explore44study.com. And as we have sites that are activated and able to enroll patients, we are posting them on clinicaltrials.gov and onto our website. So I encourage you to continue to go to the website to see when sites are activated there in Canada. So our next steps is we do plan to share our healthy volunteer safety and exon skipping data by the end of this year, which is coming up very soon. And then we will continue to enroll patients with DMD 44 and Part B. So just a little bit about the study design. Our key enrollment criteria are listed here. We are looking specifically for Duchenne patients that have mutations amenable to exon 44 skipping. And so we are confirming that through genetic testing. We do provide genetic testing as part of the study. We do have a very wide range for age inclusion. So we're enrolling boys and men that are 7 to 27 years of age. Our weight requirement is at least 23 kilograms. We're also allowing patients to either be on steroids or not on steroids. We just ask that the dose, if the patients are on steroids, it's stable, or if not on steroids, just plan to stay stable with no steroids. We're also enrolling both ambulatory and non-ambulatory patients. And then as part of the study, we do have some inclusion criteria that's related to some lab tests. We're also looking at heart and breathing functions, and then we're doing some functional assessments that are listed here, such as the 6-meter walk test and pool. Most of those functional assessments, they would be only required for those patients that are ambulatory. And then what happens during the study? The study is about 8 months in length, and that includes the screening period, the treatment period with the infusions, and the follow-up period. We are targeting doses every 6 to 8 weeks, and there's a total of 3 infusions during the study. So we're very excited about the long duration in between doses, and really hope that that does reduce burden with some of the more frequent infusions that may be required by other programs. We have various assessments that are being conducted during the study. So we are doing muscle biopsies, blood draws, lung function tests, and again, the mobility assessments. Many of those would be only for the ambulatory patients. And then we do have patient-reported and caregiver outcome questionnaires. And then we are planning an open label extension study that will follow the Explore 44 study. And the plan is for all patients to roll directly from Explore 44 into the open label extension. And in that open label extension study, all patients will receive AOC 1044. There is no placebo in the OLE study. We also have some things that we have implemented on the study to hopefully help reduce the burden on the patients and the caregivers and enhance the experience in the study. One of those is home health. So we do have a home health vendor across the United States and in Canada. And the goal is to hopefully reduce the amount of travel time and going into the clinic by having some of those visits done in the home. We also have a travel concierge vendor. And so this travel vendor will arrange all travel ahead of time. So they can book any ground transportation, airfare, lodging, and pay for that in advance so that burden is not put on the family. And we also have reimbursement through that same vendor. So, for any families that may be driving to the clinic, we do reimburse for the mileage, for parking, meals, any of those additional incidentals that occur while going to the clinic for these visits. And so just in summary, I didn't show any of our non-clinical data during this presentation, but did just want to note we do have a lot of preclinical data, including a mouse model of Duchenne, so an MDX mouse. We did see exon skipping in that mouse model, and also restoration of dystrophin protein and improvement in muscle function. So we're excited about that and hoping it translates into patients. Our trial, the Explore 44 study, is ongoing. And here it says Plan B is, or Part B is planned for this year, but we have began enrollment, as I noted, this summer. So that is ongoing. We have IV infusions every 8 to 6 weeks, or 6 to 8 weeks. And again, we're partnering with the Duchenne community and patients and families. And our goal is to make the study as burdenless as possible, and also provide additional information and resources throughout the trial as more information becomes available. So I think that was my last slide. This is our group. This was probably about 6 months ago, I would say. So we are growing and getting bigger, and we are so committed to all patients living with rare neuromuscular diseases, and especially the Duchenne patients as well. And so if you have any questions or would like to reach out to us, if you have questions about the Explore 44 study or our AOC technology or anything in general, our advocacy email is at the bottom of the screen. It's patients@aviditybio.com. So thank you again for having me. Really appreciate it.
Thank you, Trista, for presenting today, and I hope you're able to stay online for any questions that may happen after. Our next speaker is Johannes, the clinical development lead of the Duchenne program at Regenxbio. And I can see you online, and I think they'll just be getting your slides up.
Okay, thank you.
Excellent, and we can hear you well.
Okay, perfect. Thank you so much. So as the slides are getting pulled up, I just wanted to also thank you guys so much for having me here today. You're such a lovely group. I had the fortune of being there last year and apologize for not being able to make it this year, but very glad to be speaking with all of you today. I'm not sure if I see my slides. Yeah.
I think they're just working on them right now.
Okay.
We'll let you know when they're up, so then you'll know when to start.
No worries. If you need me to pull up slides on my end, I'm happy to do that as well.
No, I think they are up now. Are you able to see them?
Yes.
Perfect.
Yeah. Okay, so I'm just going to ask for next slide as we move on because I don't think I have a control. Perfect. All right, so that's me. You've already seen me here. Next slide. So I'll tell you about some forward-looking statements from our company. And sorry, we didn't go through the first initial slide here. So my name is Naz Daskir. I am the clinical development lead of the RGX-202 program at Regenxbio. I'll give you some more information about our company. These are some forward-looking statements that we usually have to present at meetings. So hopefully you've had some time to take a look at this. Next slide. So I'll tell you a little bit about our company. Next slide. So we are seeking to improve lives through the curative potential of gene therapy. We are a leader in AAV gene therapy with exclusive rights to the NAV technology platform. We have a broad internal pipeline with expertise across multiple therapeutic areas, including neuromuscular disorders. Our AAV manufacturing is proprietary. That's the NAV Express platform. And we also have internal manufacturing facilities. So if you're ever interested to know, we are based in Rockville, Maryland, and we have our manufacturing facilities right there. We have end-to-end capabilities from research to drug development and through manufacturing of drug. Thanks. It's okay. Next slide. And so what I'm going to give you today is an overview of the RGX-202 program, which is our gene therapy for Duchenne muscular dystrophy. So RGX-202 is an investigational AAV gene therapy that uses an optimized microdystrophin construct for the treatment of Duchenne. So we use AAV8 vectors. So this is an AAV8 vector. I'm sure you heard about from our other lovely companies today about AAV9 and AAVRH74. We are AAV8. And this is designed to deliver the transgene to the skeletal and heart muscles in the body. That's holding within it a microdystrophin transgene that is unique to our company. It is expected to encode a novel protein that's designed to retain the key elements of full-length dystrophin, including a C-terminal domain. So that's our unique feature. And our transgene can get to the right spot because it also uses something called an SPC512 promoter that's intended to drive the expression of RGX-202 microdystrophin in muscles. Next slide. So as I mentioned, our transgene is unique. The unique feature is the C-terminal domain. So on the left side, and I know you've seen this many times, this is a picture of the dystrophin protein as it exists. And on the right side, you'll see RGX-202 microdystrophin. So because there's only so much that you can fit into an AAV vector, we had to kind of pick and choose key parts of it that we wanted to include in there. And we were able to fit in all those features that you see here and the addition of a C-terminal domain. This dystrophin protein anchors to the sarcolemma or the cell membrane of the muscle. And we're hoping that the additional C-terminal domain kind of helps anchor it even better and improve muscle membrane stability. But obviously that's what we're testing with our human studies that are currently underway. Next slide. So our current clinical study is called Affinity Duchenne. This is a phase 1/2 study. Next slide. So here's some information about the clinical trial design. So before we even get started, we need to do some baseline assessments. This includes microdystrophin protein expression, which has to be done via muscle biopsy. We do baseline assessments of muscle strength and function. And we also perform muscle MRI and cardiac MRI. And we, as some of the other companies that discussed today, do start some immunosuppression, but a little bit more immunosuppression to kind of help prevent any potential adverse events that can come about from the use of AAV vectors. And that immune suppression regimen starts in the screening period, even before you get dosed with the drug. So we kind of get you ready. And on day 1, you receive RGX-202. So you have your baseline muscle biopsy. In order to make sure that our drug is getting to the right place, at around 3 months, we check microdystrophin protein expression again. And that's where we can see how much microdystrophin you are expressing, and meaning how is your drug getting delivered to the muscle. Subsequent to that, we then go ahead at select time points and also check muscle strength, function, and MRI outcome measures. The whole Affinity Duchenne study itself is 12 months in duration. Our primary endpoint, because this is a phase 1/2 clinical design, is to prove that the drug is safe and tolerable. We we adjusted our schedule of dosing. If you recall from our last year, we had mentioned that we would do 3 staggered doses. We are actually doing 2 now, as it relates to dose level 2. So what we're going to do is we've actually dosed 3 patients with RGX-202 to date, and we are proceeding to go ahead, and we got the okay to proceed with dose level 2. So hopefully that will be happening in the near future. And with the dose level 2, all we will have to do is dose 2 patients, one who will be less than 25 kilograms and one who will be less than 35 kilograms. And after that, we will be able to proceed to do a dose level 2 expansion. This has implications for our study because the staggered dosing does kind of slow things down a bit, and we feel comfortable with the safety profile of our drug, so that we feel like, it's okay to go ahead with the 2 doses spread apart with the weights, making sure that everything is safe as we increase the weight, because higher weight means higher vector load. So we want to make sure we're safe before we go to the dose level 2 expansion, but we think we can do that with 2 patients. So hopefully that will help us kind of complete this phase 1/2 study faster. Next slide. So I'm going to talk to you a little bit about our interim results. Next slide. So RGX-202 was well tolerated with no serious adverse events in 3 patients. So there was a data cutoff at the end of September. So I'm a formal data cutoff at that point. So I'm only going to present you data from that point on, from that point to prior. So just so you're aware. So we're a little bit beyond in terms of time post-administration than what's listed here, but we're still doing okay. So we have patient number 1, who was 4.4 years old at dosing, weighing 17.8 kilograms. And we had 6 months of data from that patient that we can share with you. Patient number 2 was 10.6 years at dosing, weighing 28.3 kilograms. And we have post 19 weeks data to show you. And patient number 3 was 6.3 years at dosing, 26.8 kilograms. and 3 weeks after dosing. So 3 weeks after dosing isn't going to give you any microdystrophin expression yet. If you recall from our prior slide, we got the microdystrophin from the muscle biopsy at 3 months, but we'll be able to show you some info on our other patients. Next slide. So here is the phase 1 interim data from our study. So RGX-202 microdystrophin expression was observed at 3 months with comparable results obtained via Western blot and liquid chromatography mass spectroscopy. It's just another way of checking for microdystrophin expression. We did notice that the microdystrophin expression at 3 months in our first patient was 38.8% of the normal control. Our CK levels also decreased from an average baseline of 15,278 to 8,692. Obviously, this is early data. Our last recorded measurement at the data cut was week 10, but we'll continue to monitor this as well over time. Next slide. So for patient 2, we have the 10-year-old. So first patient was 4.4, second patient is 10.6, who was 28.3 kilograms. The percent microdystrophin that was measured on the biopsy was 11.1%. And the CK levels dropped from an average baseline of 17,437 to 9,756. Next slide. Here's a sample of the images that we have of the RGX-202 microdystrophin immunostaining from a healthy control, an unaffected patient at baseline, and then the baseline for our first patient, and then 3 months after RGX-202 administration. So what we're looking for is this bright red staining that you can see in the normal tissue here. The bright red staining that you're seeing is lining the muscle fibers, all of those little polygonal shaped muscle fibers there, and they're tracing the whole edge there, the periphery. So that's someone who doesn't have Duchenne. When you go to our baseline, you'll see that that bright redness for our first patient is not noted here. And also there's like a lot of variability in the muscle fiber size. When you go on to the 3-month evaluation, you can see that there's much more immune staining of microdystrophin along the muscle membrane as compared to the baseline. Next slide. So a summary is that the RGX-202 at first dose level, so we had to start with the first dose level, which is our lowest effective dose. So obviously we're seeing benefit from this dose, but we do want to see, again, from what we saw in animal studies, we do want to see what we can gain with an even higher dose. And that's with dose level 2, but dose level 2 can also present with additional risks. So we're being very cautious about that, and we're watching it very carefully, but we're very pleased with the safety results we've seen from the first dose level. It was well tolerated in 3 patients with no serious adverse events. We readily detected RGX-202 microdystrophin expression in the 2 patients at dose level 1 at the 3-month visit. The RGX-202 microdystrophin localization with that bright red line that we saw in the sarcolemma supports the distribution that we'd like to see in muscle tissue, and the CKs also dropped after RGX-202 administration. So we're hoping that that's also improving sarcolemmal stability. Next slide. So, we're accelerating clinical development. Our dose escalation, dose level 1 interim results, and new preclinical data support immediate escalation to dose level 2. Our enrollment is expected for dose level 2 is expected to initiate by the end of this year. We expect to make pivotal dose determination and initiate our pivotal program in 2024. For an accelerated approval, we expect to file a BLA using accelerated approval pathway with microdystrophin as a surrogate endpoint for clinical benefit. I know Dr. Hoffman was talking about that earlier. That's to kind of get us going, but we're also hopefully going to be showing some functional data along with that, and have commercial ready product, which is material from Regenxbio's manufacturing and innovation center, ready for dose escalation expansion. It's a process that's in place for our pivotal program and ready to distribute, hopefully once we become commercially available. Next slide. So this is our lovely team. A lot of really great people working at Regenx. I'm very happy to be a part of this team, very dedicated to the Duchenne community. There's a lot of heart behind where we're coming from, and we're so pleased to be working for such a wonderful group as patients with Duchenne, and we're hoping to be able to bring more great products forward for the community. Next slide. If you'd like to learn more about Regenxbio, this is our website. We welcome you to stay in touch with us at Duchenne@regenxbio.com, and this is our clinical trial listing for Affinity Duchenne. Thank you.
Thank you, Naz. We're holding our questions until the end. And so I'd like to introduce our last speaker, Catherine. She is the Director of Patient Advocacy at Edgewise Therapeutics.
Hi, everybody. I'm so excited to be here in the room with everybody. As mentioned, I'm the Director of Patient Advocacy at Edgewise Therapeutics, and we're actually based in the United States in Boulder, Colorado. And I'm here today to talk to you about EDG-5506. I'll say that a lot. It's our investigational compound, which is taking a novel approach to protect muscle in Becker and Duchenne muscular dystrophy. Another forward-looking statement slide. The main takeaway here is that we're a public company, and I may speak about things that may happen in the future, and that EDG-5506 is an investigational agent and is not approved in any territory. So Edgewise was founded in 2017 with the aim to improve lives of individuals living with rare muscle disorders. We are developing a novel mechanism to protect the muscle in Becker and Duchenne muscular dystrophy. And we believe that our compound has the potential, it's a daily oral pill, and has the potential to be used alone or in combination with other therapies, regardless of mutation. So this is a non-mutation specific therapy. So we have a few. Even since the submission of these slides, we actually have more clinical trials than this ongoing. Our first was ARCH, which is a 24-month open label study, which is ongoing in adults living with Becker. And that was a phase 1 trial. And then Canyon, which is a study in ambulatory adolescents and adult males with Becker, which actually just finished enrollment. And we announced Grand Canyon, which is a pivotal study in adults with Becker, which will be a global study. And then Lynx, which is our initial study in ambulatory boys with Duchenne, which is also just finishing enrolling. And not on this slide is Fox, which is our most recently announced study, which is a study in boys who have been previously treated with gene therapy, where we will also then be studying the effects of EDG-5506 on those that have been treated with gene therapy. So what is our approach? Well, Edgewise, our approach is to protect susceptible muscle fibers. So we know that some muscle fibers are more susceptible in individuals with muscular dystrophy, and these fibers are susceptible to damage due to that lack of functional dystrophin. So we've made an investigational compound, EDG-5506, that's designed to protect these fibers from the damage regardless of mutation. And in diseased animal models, EDG-5506 protected those susceptible muscle fibers and prevented that long-term development of damage. And then in recent, we've been able to report out some of our clinical data. So in clinical trials, we've actually seen a reduction in biomarkers. What's a biomarker? Biomarker, in the simplest terms, is essentially a way to gauge abnormal or normal response when proteins leak out into the bloodstream. So we've seen a reduction in two things when we're looking for muscle CK, which most of you are aware of because it's a good diagnostic tool for Duchenne, but then also fast skeletal muscle troponin. So we've seen a significant reduction in the biomarkers that are related to muscle damage. And then we've also seen a trend towards functional improvement in Becker. So let's back up. How does dystrophic muscle break down? Well, dystrophin here, as you can see on this schematic, is the green, and it's anchoring the fibers. So it connects all the contractile proteins to the muscle membrane in these fibers and that surrounding matrix. So a lot of us think, okay, we're contracting our muscle, that's all happening at the same time, but really you're getting signals sent to individual fibers and they're contracting at different rates and different times. So dystrophin helps to stabilize that, as you can see here. It's spreading the force across the fibers. Well, when dystrophin isn't present, that support is lacking. So we'll see that here in the schematic, that when we take dystrophin away and we have the muscle contraction, that then all of that force is being sent to a singular fiber here, as shown in the schematic in the middle. And that's where we see damage occurring, the root of the problem to damage in muscle in Becker and Duchenne. So let's take a peek in action. So this is actually a finger muscle of an MDX mouse. So on the left, which a finger muscle is thin enough that we can actually look through it on the microscope and see the contraction occurring. So on the left side, we see that this is a non-treated dystrophic mouse model, and the finger is the muscle is contracting, and you see those little blobs that are forming. In the most elegant terms, the blobs that are forming is actually the fibers breaking down, and that's the damage. We see those visible changes, that contraction induced damage, and all of that flooding of the damage that's occurring. And then on the right side, we've treated this with EDG 5506, our compound, and you see that during contraction, we're not seeing that damage. So we're altering the way that a protein called myosin, we're altering how muscle contraction occurs. It still is able to function, but we're not seeing the damage that's normally attributed to what we see in daily activity through contraction in Becker and Duchenne. Maybe it will move. Okay, so let's talk about Duchenne trials. If you have questions, I know we had a few Becker questions yesterday come through. Happy to give more information about anyone that's interested in the Becker trials. But for this group today, we'll focus in on our Duchenne trial. So we initiated our phase 2 trial in boys with Duchenne at the beginning of 2023, and we've enrolled over 30 individuals with a genetic diagnosis of Duchenne. These boys were ages 4 to 9, ambulatory, and met certain functional criteria. And these individuals were able to be either on a stable dose of corticosteroids or on an approved exon skipping therapy. And this is a 12-month trial, which had a 3-month placebo period, which meant for 3 months, there were certain individuals that were not receiving the active study drug, rather the placebo. And then after that, everybody is moved to a 9-month open label. So again, open label means everyone's receiving the study drug for a 9-month time period. It's a relatively short time period, 12 months, but really what we're trying to aim to understand here is what dose is best to take forward into a longer trial. So this year-long 2-part study really is hoping to understand the safety of the drug, and again, what dose is going to best be used so we can move forward in a phase 3 design for the trial. So there were approximately 10 site visits over the 12 months. We had some at-home elements included for blood draws to be able to monitor the boys and reduce the number of times people were having to visit sites. And then there were select functional assessments throughout this relative to the North Star ambulatory assessment that most are doing in clinic anyways, but blood tests as well, and no muscle biopsy. So in summary, EDG 5506 is being developed for Becker and Duchenne muscular dystrophy. It's a daily oral pill intended to preserve and improve function in both Becker and Duchenne. But really, our goal is to protect damage to the muscle by protecting those most susceptible muscle fibers, and potential to be used alone and in combination with other therapeutic approaches across the dystrophinopathies, and really stopping that damage where it begins when it relates to the muscle. And that is really all I have for everybody today. But if you want to get in contact with us, I'm here, so come ask me questions. But also you can reach directly out to our studies team at this email or find us on social. But really thankful for Defeat Duchenne Canada and for all of you and your attendance. So looking forward to connecting. Thank you.
So I'd like to thank all of our industry sponsors for speaking today, and specifically for this section, Alana, Trista, Naz, and Catherine. We're going to move on to questions. I don't know.
We have some
Mhm.
online questions. for this
Okay,
session
perfect.
there. Starting with Edgewise, can you just touch on plans for clinical trials in children with Becker in Canada?
Oh, wonderful. Okay. I'm pretty loud, but we have to go online. So right now, so we just finished our adolescents enrollments, which was in Canyon. And the next phase of this, the pivotal cohort, is only going to be in adults. So at this time, hopefully the pivotal will allow us to move forward with an application. So we've already enrolled for our adolescent portion, and there won't be any more adolescents enrolled moving forward. And we hope that that means that we'll move forward in our process for application as well.
Great. Any questions in the room?
It could be the NOx studies for like post gene therapy. Like I was wondering about exon skipping. Would it be used because it's it's for protecting damage to the muscle? So could you use it also in combination with an exon skipping?
Yeah, so we're actually in our trial for Duchenne allowing individuals on approved exon skipping therapies to be in our trial. So yes, we're hoping to be used in combination with a variety of different approaches. Yeah.
And I just want to make one comment. So right now, we don't have exon skipping medications approved in Canada.
Mhm.
So I think we would need to just be thoughtful about that and understand what that would look like if
Mhm.
there are Duchenne studies for this medication in Canada, and understanding where we are in Canada with from an approval process.
Great point.
All right. I think if there aren't any other questions, I just really want to thank once again everyone that has been speaking today from our industry partners. And thank you everyone that was able to join online as well.
So again, thank you, Dr. McAdam, and thank you to all of our industry partners for providing these informational sessions today and the latest DMD research and drug developments at your respective companies. So now this morning, we've heard the latest in research, but how can we bring these treatments to Canada to ensure our children and young men have access? That's where advocacy comes in, and we hope some of you will be able to join us for the remainder of the weekend and tomorrow on Capitol Hill as we press for access to affordable treatments in Canada. Excuse me. As we conclude the 2023 Family Forum, I want to say a huge thank you to all of our speakers for joining us in person and online. And a huge thank you to you, our families and supporters here in Ottawa and virtually across Canada for engaging in this annual event. Again, a big thank you to our partner, the Children's Hospital of Eastern Ontario, and Dr. Leanne Ward for helping us coordinate such a great agenda. And to our amazing educational sponsors, we wouldn't have been able to offer such a world-class educational opportunity for all of us at no cost to our Duchenne community without your support. Again, personally for me, it was an honor to be your host, MC this weekend, and I want to pass it over to our new fantastic CEO, Lisa. Come on up, Lisa.
Thank you, Brian. So of course, I echo everything that Brian has said. It's just been a phenomenal weekend of learning and engaging. So thank you to everyone who's participated and to all our wonderful speakers. It's been tremendous. So as you can imagine, it's no small feat to put an event like this together. So I just do want to take a minute to give a little shout out to our team at Duchenne Canada, Rick Moss and Kate Foster, who's over there, and Rochelle Tenhalf. These individuals have worked for months to put this together. And in addition to that, all weekend, we've had Allison Boyce with us and Allison Davidson, who have just been rolling up their sleeves and helping out in many ways throughout the weekend. So maybe we can give all of them a bit of a round of applause. And I really didn't have to do anything, so I don't know if I'm going to be able to keep that going in future years, but they did a phenomenal job. As all of you know, ever since John and Jesse walked across Ontario in 1995, this organization has been about all of you, our community of families, parents, caregivers, clinicians, researchers, supporters, and most importantly, the young people living with Duchenne. And this is what we will ensure continues long into the future. We had the opportunity to engage and meet a few of those young people who were able to attend in person today, which was wonderful. We thank them. We know there were many more across the country and many more families across the country that we engage. But I wanted to just take a minute to right now and introduce you to one of them, Emery. He's coming. Believe me. Okay. So let me tell you a little bit about how Emery is going to be helping us. Well, first of all, let's just say that everyone knows, both those in the room and of course online, that Duchenne is a disease that impacts the entire family. And this is why, as an organization, we've always prioritized the development of resources like our family forum that bring hope and knowledge and courage. And starting next week, we are launching a campaign, and every dollar that's donated to our charity will go twice as far. Every dollar that's donated before the end of this year, December 31st, will be matched dollar for dollar up to $65,000, thanks to an incredibly generous group of donors. Emery will be featured in this campaign, and we thank Emery and his family for doing that. So please keep an eye on your mailbox and your social media and your inbox to help share Emery's story and to raise funds for Duchenne research, care, and advocacy. We're really thinking this is going to be a tremendous campaign, so we thank you all in advance for your support. Please remember to keep up with our educational opportunities and resources by signing up for our e-newsletter. Follow us on Facebook, Twitter, Instagram, LinkedIn, YouTube, etc., as I'm sure many of you do, at Defeat Duchenne. And please mark your calendars now, save the date for our virtual walk to Defeat Duchenne, which is scheduled for the 26th of May, 2024. Remember to visit our website for more resources. And as we heard this morning, and as Brian mentioned, now is the time for Canadians to join the fight to defeat Duchenne. And we will be working with our advocacy ambassadors this afternoon as we begin to prepare for our advocacy summit and some impactful meetings that we'll have with our elected officials tomorrow. So thank you all again. Thank you for giving up your weekend to be with us in person. And for those who've joined us online, and I know there's many of you, we really appreciate you doing that. I'm sure that you have found this weekend as informative and inspiring as I have. And we look forward to doing this again next year. So thank you all and safe travels.